I think so.
All right, we're recording.
All right. I'm going to share my screen again. And we're going to do a little bit more antimony elements and pieces and things like that. As you may recall, so during the break, I redid this. So we don't need to rerun that again. We've talked about rate rules, assignment rules, and I thought I would tell you a little bit about compartments, especially because that model we looked at just now had compartments. So SPML and antimony allow you to have species in particular content compartments. So basically they say that like there is a region with stuff in it, and then there's a different region with different stuff in it. And if the same thing, like if you have ATP in the extracellular and in the cellular compartments, then they are officially different quote unquote species in this parlance. And then you can annotate them as being the same thing, as being ATP officially, but ATP outside and ATP inside are officially different species in all our modeling and in all of these types of models as well. So let's say this is our S1 to S2, S2 to S3 model. Let's say that S1 species S1 is in compartment 1, and S2 is in compartment 2, and S3 is also in compartment 2. And then I can initialize C1 equals 1 and C2 is also size 1, and that won't change things at all, right? We still get exactly the same simulation, because even though S1, essentially this one is S1 is being imported into a cell, say, and becoming S2 at a particular rate. So now let's say that our compartment sizes are different. That's the whole reason why you have compartment modeling is if the compartment sizes are different or if they change in time or something. So now we'll say that compartment 1 is size 10 and compartment 2 is size 1. So the first thing to do is to like think about, what do you think will happen? The rate at which S1 goes to S2 is based on the concentration of C1, but then it gets, but these are elements. This is the amount of S1 becomes a certain amount of S2, and and it comes in at a particular rate, but now it is in a much smaller compartment. So if we simulate this, we get a much different reaction here. And let's, here, 0, 30 or something. So S2 goes way up, much higher than S1 was originally. And S3 then goes like super high up to like 40 here. And as you might, if we go, let's do the steady state thing. Oops, steady state. And just look at S3. And as indeed, when you take this to steady state, S3 is 50 now. And that's because we had a concentration of C1 that was the concentration was 5, right? But that was based in the first compartment. And if we are forcing all of those species into a much smaller compartment, the total amount of species of S1 is the same, but now since it's in such a smaller area, it has a much higher concentration. Now, if I were to say selections equal time, and then just S1, S2, and S3 like this, that is going to show us our old reaction. And then we're going out to 30. So let's go back to, were we at 10 or something? No. Oh, just 5. Anyway, so now, okay, so S in a selection list, just S1 doesn't mean the concentration of S1, it means the amount of S1. So now we're plotting the amount, and the amount starts at 50. So here, we'll go back to 50 here because it makes more sense. Shows you a little bit more of the thing. So now we can see the conservation of mass because we're plotting mass instead of concentration. If we want to look at the concentrations again in this format, we want to put them in brackets like that. So now we're looking at concentrations, and now instead of S1 starting at 50, it started at 5 and is going down, and the other two are the same as they were because their compartment size is 1. So the question there, are they independent in this example? So the answer is no, there are things transferring between them, and that is determined from this reaction right here, right? We had S1 in compartment 1 becoming S2 in compartment 2. And these reactions happen at a rate based, all reactions are based on, officially the term is extent, but it's basically like the amount of S1 and the amount of S2 in any given click is the same. So if we had a lot more literal levels of S1, then it would become a lot more S2. If we, let's see here. Let's have C1 change in time. And let's go back. Yeah, these are our default things. And in fact, so I'm going to change this I'm going to change this more. Nope, I'm going to change this. So now we have C1 at 1. It's going into a larger compartment, but that compartment is slowly getting smaller. Oh, and it didn't work. Let's see here. Let's just make more time points. That's still not working. All right, this got too complicated and it didn't work at all. So what would happen here if I just comment that out? Does that work? Yeah, okay. So this is sort of I've been making things up as I go along, and the model itself got unstable and is highly dependent on very small changes in the math, and it's too small to be accurate in that case when I was having it changing in time. So I don't know. Okay, here we'll do it the other way around, and we'll say that the change in time, so C2 is getting larger. There. And for some reason, it's showing me C2 on this thing. No. Why are we showing me C2? All right. I'm 2, 3. There. All right. Why did it not like steady state? Oh, it failed to converge to a steady state. Yeah, because things are changing in time, so that doesn't work. In that case, so we failed our steady state call because C2 is just constantly changing. So there is no solution to the steady state problem because everything, there's nothing that regulates the change in C2. So if that happens in general, that will just, your steady state will fail. So yeah, now we have our second, we start from compartments of size 1, but now compartment 2 is getting larger and larger. And it's taking longer for the change to happen. So we immediately went up to make S2, but then it's taking a lot more and more time to convert S2 to S3 because the compartment's getting larger and the molecules can't find each other basically, right? It's the rate at which this happens is based on, by default, the concentration of S2 not the amount of S2. So, oh, if you done, speak a little bit more about the logic, the compartment size, and how that changes things. So let's see here. So if you have a compartment that is changing size, that only changes the solution, basically, like the part that is not modeled and the same amount happens. In fact, let's just say, let's make this much simpler. Let's comment out the reactions entirely, right? And we'll say 6 and 8 or something, I don't know. So now the only thing that is changing in this model is the compartment size. And of course, since I told it to plot the amount is not changing either. So we'll go back to this. And if we plot concentrations, then the concentrations are changing in time because the volume in which the same number of things are is making it larger. So yeah, the compartment size it does have a unit. It has a volume. So the volume if the volume is changing and the amount is not, then the concentration, which is the amount over the volume, is going to decrease. Does that make sense? Yeah. So like this would be what would be happening in the background, and then if this is also happening, then that's what happens. The fact that I sort of overwhelmed this with the initial bits. Yeah, now we have S1 like decreasing quickly, and yeah, this time the C2 was changing anyway. And yeah, I'm like this reaction from different compartments may not make as much sense. Like mass action isn't necessarily the best way to model that sort of change. But that is just what my model said. My model said that S2 is imported at a rate of K1 times S1. And it didn't, and S1 by default is a concentration. So as things go in, sort of change compartments, this reaction rate determines how that happens. And if there are probably more realistic models for more realistic equations that show like how osmosis works or something. Yeah. So that's a little bit about compartments. Compartments can change. They can be non-size one. If you don't say anything about compartments, Antimony will actually create. In fact, let's do this real quick.
There's a bunch of questions on chat about compartments.
May I get a clarifying question here regarding the concentrations? So I guess I was getting a little bit confused. If you're saying S1 is a value of 5, is that saying it's 5 units of whatever it is you're talking about, not actually a concentration until you've given it a compartment size?
No, it's a concentration.
It is by default S1. If you just say S1 equals 5, that's a concentration. If you look at this is our original model, and you'll see like in the SML, it has an initial concentration of 5 here. And
but I guess though, if you have a concentration that is, if you're changing the volume, that shouldn't change then, should it? Because it's a concentration. But if you're adding these compartment details, then are you telling it to adjust the concentration according to the volume that you're creating?
So,
because a concentration.
Yes. So in SVML, if I say the compartment volume changes, then I am saying that the solute is changing in time, and not that like you can imagine like pouring in, like I have S1 in there and I'm pouring in a solution that also has S1 in it, right? But I would have to model that differently. by default, if I say the compartment volume is changing, if I'm like pouring something in and making the volume larger, I am only changing the size. I am only changing the solute. I'm pouring in deionized water or whatever, right?
Okay.
So the concentration. So if I pour in deionized water into a particular thing, then the concentration of everything in there is going to decrease because the volume is increasing.
The other thing I should point out,
Mhm.
internally, the simulator works with amounts because
Right.
it knows that the concentration, the volumes could potentially change. And so in working with amounts, whenever it needs a concentration, for example, in a rate law, it'll take the amount and divide by the current volume. Right? So it's actually working in amounts internally all the time.
Okay, that makes more sense to me. So
yeah.
is there a default volume that it's operating from if you
Yes.
don't give it compartment size?
Right.
One.
So the default
One. Okay.
default compartment will be a size 1 here.
Gotcha.
Right.
Thanks. That helps clarify.
Yeah.
So that also because we work in amounts, that also ensures strict mass conservation.
Right.
Yeah.
So if your model otherwise is as mass conservation and isn't just like S1 goes to nothing, then then you can change the compartment size and it still works. That's actually another reason to model things in terms of reactions instead of like just rate laws. If I had put this in terms of just rate laws, then changing the concentration wouldn't have changed. It wouldn't have affected things correctly, basically. Does that make more sense? This is indeed a somewhat confusing topic, but yes. So reactions always work by changing amounts. The rate at which they change can change in terms of concentrations. And you can actually say, I'm going to forget the keyword, but here I'm, yeah. That's one. If I say this, and then if I get S1, oh, yes. Nope. Ah, here we go. Has only substance units is true now, right? And by default, it was false. And that means that officially, when it appears in math anywhere, in reaction rates or rate laws or assignment rules or anywhere, it is the amount that is being used in that math and not the concentration. I'll leave it at that. If anyone is dealing with things that you have to have the substance only, just know that you can do it with that keyword. Also, if I say in C1, and if I initialize it and say that is sort of antimony, that's telling antimony by default that is like, oh wait, 5 is actually not an initial concentration, but an initial amount. So you can now see S2 and S3 have an initial concentration, and S1 has an initial amount officially, because I said it was 5 divided by C1.
So another question, can S1 be in 2 compartments at once? No.
Right.
What you would do is you would name them differently. So you'd say ATP cytoplasm and ATP mitochondria. So they'd be treated as different substances, but in your mind, you know they're the same, but just in different compartments.
Right. So the definition in this kind of modeling of a quote unquote species is a thing in a space, a thing well distributed in a particular space. So if it's ATP in the mitochondria, then there's ATP well distributed, well mixed in that mitochondria. And if you also have ATP in the cytoplasm, that is a different thing. That is a different quote unquote species, because you treat them as separate. You give them separate IDs, ATP. So yeah, you could say like this is ATP-C goes to ATP and something, right? Like that. Yeah. So that's that here. I'm going to comment this.
I mean, this kind of formalism we have here, it's particularly good if you're doing compartmental models. There was a question about osmosis. I can't remember how to deal with osmosis because that's a movement of the solute itself, of the solvent. I'd have to look into that. I don't know how you deal with that. But there would be a way. I suspect it's modifying the rate law in some way.
So yeah.
Yeah.
So now we have sort of the is keyword and this just sort of gives human readable names for these things. And like in the SVML, if we look at them, what's the if you get this gives it a name: ATP in the cytoplasm, ATP in the nucleus, and in some in some plotting it'll grab the name for you instead of anything else. So anyway, all right, compartments. We talked about distributions. I actually did talk about distributions the other day, so I'll skip those for now. I talked a little bit about events. Let me use that for something else. Events. Oh, this doesn't even have my event example. Okay, I'm going to talk about events in this. So we have this. This is our standard model, right? And we want to say, I told you yesterday that you could say at S1 is less than 3, S1 equals 5 again, right? And now every time it gets less than 3, it bumps up to 5. You can, oh, and someone, there are, so you can also set multiple things and say, say S2 equals 0. You can say like both of those happen at the same time, right? So now S2 goes up and S1 comes down, but then they get reset. But S3 never gets reset, so it slowly is creeping up over time. And yes, you can use time for events. You can say at time is greater than 3, S3 equals 3 or something. So it was creeping up, and then we just hard set it to 3, and then it keeps on creeping up there. So someone asked another question about compartments. What's the main advantage of using compartments? The main advantage of using compartments is that if your system involves multiple compartments and those compartments change in time, or you're dealing with concentrations, but the import to the export is going to change those concentrations. Talking about it in terms of compartments, from a small compartment to a large compartment, can sort of get all of that math for free, and you don't have to worry about it anymore. If you just say ATP in the mitochondria is exported to the cytoplasm, then you just say that happens, and then the concentrations just sort of work out regardless of anything else. It's just an ATP molecule is ported out, and it changes concentration, and off you go. You'll notice that I did not say at time equals 3, because that might not work. It did work, but it might not have. And the reason is because
why have you got 2 equals?
Because equals means 1 equals means assignment, and 2 is a test.
Okay. Not everybody might know that.
Yeah, that's a fair thing. So and equals is a check to see if those 2 things are equal to each other. So internally, the simulator is going on, and it's taking steps in time, and it's like, okay, and then it goes to check to see if any of the event triggers, if these sorts of things, is S1 now greater than 3, is time now equal to 3. And the problem is, if it was at time slightly less than 3, and then it advances and it's slightly more than 3, is like, did that trigger change? And it was 2.9. Is 2.9 equal to 3? No. Is 3.1 equal to 3? No. Was there a change? No, it doesn't realize that there was a change from 2.9 to 3.1 because it didn't check the exact moment where time was exactly 3, and it sort of like blipped up to true and then went right immediately back to false. So to make things easier on the simulator and on the algorithm underneath, we just say is at time greater than 3, or you can even say greater than or equal to 3 if you want that, if you still want it to happen at time 3. And then when it checks at time 3.1, it knows that it has changed, and it can be like, oh, okay, I should back up my simulation to when it wasn't. There's a thing called a root finder. It goes back to find the exact moment at which that trigger changed, and then it can proceed accordingly. I promised I would talk about piecewise functions, so I'm going to do that. Let's see here. I'm going to take out this. So I'm going to have this one, but instead of K1 equals 0.6, I'm going to say it's equal to a piecewise function. So piecewise functions mean that a particular is a mathematical equation that returns different values depending on different conditions. And the first thing you do is a first value one. So you say val1, cond1, val2, cond2, etc. Otherwise, right here, I'm going to comment that out. Where did I go? Oops. Okay. Copy, paste. All right. So I want the piecewise. So we had it at 0.6. It was 0.6 as long as time is less than 1. And then if time is not less than 1, we want it to be 0.8 as long as time is less than 3. And then otherwise, we want it to equal to 1.2. So this is sort of stepping up K1 in time. So it starts off at 0.6. After time 0.1, it jumps to 0.8, and then it's going to jump again to 1.2. And if we do that, it's hard to see what's going on because here, maybe I should do like 1.8 and 8.2 or something. Oh, this is just K1. Okay, here, we're going to do K2 is this. Oh, and no, actually, the problem might have been this. I have to comment that out. Nope. Okay, we'll make it K2. Oh, crap. Nope. That was the problem. I said it was equal to. That only gives it an initial value as opposed to a. Okay. Okay, this is now an assignment rule because now that needs to apply for the entire simulation, not just at time 0. If I just say S1 equals 5, S1 isn't always equal to 5. S1 is just 5 at the beginning. So now K1 is changing in time. There we go. Now you can actually see things change. And so indeed, at time 1, it changes to go faster. And then at time 3, it went a little faster, but S1 was so low that we didn't have nearly as much of a change. So if we say that this is for K2 instead, that symbol is a comment symbol. We'll see that happening. And now we see that like it was going up, and then suddenly like it was being converted to S3 at a much faster rate, and it just sort of tore it down. And then it like converted to go even faster, and it just scooped down to 0 almost instantly.
There was one question which I answered, but just to make sure everybody understands, these transitions, these checks, we call them, sort of thresholds. When the threshold happens, that event only occurs at that moment. It doesn't occur continuously then after that.
right? So yeah, the events happen when the trigger transitions from false to true, when it becomes true. And then if it was true and it stays true, nothing happens. And if it was true and it changes to false, nothing happens. And then if it changes from false to true again, then it triggers again. Yes, someone asked if this could describe like a compensatory response, right? You can also say, like, I don't know, like if you wanted to turn off part of your model for a certain amount of time, we could say that let's do that. Let's say this only happens, 1.8. And then, so we only want S2 to S3 to happen between time 1 and time 3. Otherwise, if earlier it's 0 and after it's 0. So here at time 1, we start converting S2 to S3, and then S3 converts, and then we stop again, and the remaining S1 continues to change to S2, and S3 just stays where it was because now that it's off again.
But what you're doing is you're turning a reaction on and off.
Right.
Right. So you could use this to turn on whole batteries, whole sections of your model on at a certain time, then turn it off at another time. So you could do all sorts of tricks with this.
Right. And I can do this exact thing. So I can do it with a piecewise thing. I can also do this with events. I can say K2 equals 0 at time is greater than 1. K2 equals 1.8 at time is greater than 3. K2 equals 0, right? Now I can comment this out, and I should get exactly the same function. So this is probably slightly less work on the simulator. The simulator doesn't have to constantly calculate the result of this formula. In this case, it doesn't really matter. It does have to always check these times, but it always has to check the times anyway because they're in piecewise. So that is just as expensive as it was. But this is sort of constantly checked, whereas this is you are only checking this part.
By the way, those of you who build gene regulatory networks, you could actually probably use this to turn genes on and off. You could probably build a sort of Boolean gene network using this technique, I suspect.
Yeah.
Yeah.
And someone asked, like, could you use other species here?
Can you have Boolean operators if time is greater than 1 and S2 is less than 3 or something?
Yeah, and I think you have to use this. So S2 is less than S1, say. So this will still actually work, right? So we still get that. But if we said that it had to be, if we said it had to be greater than S1, then it's going to not trigger until that crosses, and then it's going to turn on. And let's see if that's not here. That's not enough time points. 0, 5, 100 or something. There. Now at the point where it hits that, then suddenly we've turned on our second reaction. And then we actually turned it on again here too. Look at that. Because we turned it off. Yeah. So this one, so we said at time 3 K2 equals 0. So great, it turned off again. It started to accumulate, and then it got greater than S1, and this thing triggered again, and we set it back to 1.8, and it went back down again. So,
hold on,
yes.
Hold on, I don't understand that. I thought time greater than 1 would only trigger once.
It's still true, right?
Oh, so it's always true, of course. Yes,
right.
True and true.
Right.
Yes.
So this whole condition was false at the beginning. It transitioned to this is time after 1. So the first thing that happened was time was greater than 1, but S2 was not greater than S1, so it was still false. Slightly greater than 1, both of these were true, and so the condition as a whole became true, and it fired, and it triggered K2 to 1.8, and we started consuming S2. Then we set K2 to 0 at time 3, and we started increasing it. So this part of the equation, time is greater than 1, was still true. And finally, when we got to 3.3 or something, S2 was suddenly greater than S1 again, and we set K2 to 1.8.
But you got a piecewise question. I'm not super familiar with the piecewise, so you might have to answer that question, Lucian.
Function has still behaviors with the function being smooth. Oh yeah, like, so you are correct. You can do both. If the function is a smooth behavior, then you can do both one. I would say like, if this was 6 months ago, I would suggest doing the sum function because the piecewise wasn't working all that great in Roadrunner. But then I've fixed it, and so now we actually are. It treats piecewise more like events than it used to, so it can notice those transitions much more easily. And as long as there's more than just sort of momentary things, that didn't make much sense. When I said time equals 3 as opposed to time is greater than 3, I was easier on things. And the same is true in piecewise equations. I could say time equals 1, right? And that would be useless. No, here, time is greater than 1. I could say, like, when time is exactly equal to 3, it equals 1.8, and then it would be you would set K2 to 1.8 for an instance in time, and then it would turn right back off again, and so that wouldn't be useful. But it also isn't useful because this is really an unstable thing to find for a simulator. It's hard to oops. Yeah, sure. No, I don't remember. Anyway, is this still giving us? Oh, I commented it out anyway. So yeah. All right. So yes, you can do either one. If the only difference might be in the amount of time it takes to do the calculation, and in that case, you could try them both in a really large simulation and see if it makes a difference. That might be enough. What else did I have in here? Modularity. Modularity is nice. It's sort of a large topic.
Yeah, I'm not sure how important it is.
Yeah, I will say, like, I'll just say you can use Antimony to stitch models together. I won't show you how. If you are interested, you can ask me afterwards or look at the documentation. But if you want to, like, pull in two models, like someone has this part of glycolysis and someone else has a related thing, and you want to stitch them together, you can do that in Antimony and say, like, they are stitched together this way. So it's possible. The syntax is relatively straightforward for that. Certainly easier to do than in raw SVML. Yeah, that's that.
Okay.
We have a couple, like, oh, I didn't put in the link for Antimony Web Editor, but anyway.
Yeah, events are really quite flexible. I mean, I haven't thought too much about modeling gene networks, but I think you could model gene regulatory networks. So let's say you have a metabolic pathway or a signaling pathway. That would be modeled as a set of differential equations, okay? And then on top of that, you have a gene regulatory network that turns on various things on and off, depending on the inputs, and you could model that part with a whole set of event transitions. And so you could have a hybrid continuous model using differential equations, and then a discrete model on top of that using events.
Yeah.
I suspect it's rich enough to be able to do quite a lot with that.
Yeah. And I haven't really talked about all the details with events. There's whole things with, like, if you have two events firing at the same time, there's delayed events, like you have something that triggers and it's like, all right, in 5 seconds, do the thing. In fact, I could show you that.
Oh yeah, you may also show that one. Yeah,
that actually seems somewhat useful.
I've seen people use the events to do some very interesting tricks to generate different input signals, like sawtooth, square wave, and so on.
Let's try that. Right. Okay. So now, so this one I said at 2 after, then we turn that one on, right? So now, as soon as S1 was greater than S2, this part became true. But then I said, like, I don't want you to change anything yet. I want you to wait another 2 seconds and then set K2. So it waited another 2 seconds and then turned it on. So if we
So that's definitely useful in a gene network, because when a gene turns on, it takes time to turn on. So you might want to add a delay.
Yeah.
So when the signal first signals the gene, there will be a delay before the gene comes on, and that could be modeled using this.
Right. You can also say that it has to, I feel like this should have triggered again. But anyway. This word greater than that, and this one is greater than S2, and it wasn't true here. Oh, it did, right. Because so it did trigger here, and then it waited 2 seconds, and then it turned on again, because I forgot about the delay. Make it a little smoother. There we go. So yeah, so it transitioned, it waited 2 seconds, it turned on. It went below, it started going up again, it triggered, it waited 2 seconds, and then went on again. You can also say, like, for some things only like, say you're modeling neurons or something, and you have to have a certain level for a certain amount of time, you can use the keyword persistent in your model. You can say, like, persistent equals true. That means that for the whole 1 second it's waiting, this condition has to continuously be true that whole time, otherwise it won't do it. And this one obviously is just, it's like, yes, S1 is still greater than S2. We don't care about that. But in other situations where it thinks something's going up and down, this one would say, like, I have to have a persistent signal for a whole second before I turn on K2.
So could you have
Mhm.
would that apply, say, if you had K2 equals 1.8, if you had K2 equals 1.8 times time, does that mean that?
Oh, so yes, I mean, you can put any math in there. The delay thing means that you either use the numbers from when it triggered, or you use the numbers that as true. Sure, here equals, we'll say it equals time. And then we're going to take this one off.
Oh, I see.
Right?
Yeah.
So if we do this, and then we're going to say, like, what is K2, right? So in this, K2 is 1.1. So at 1.1, it triggered, it saved that value of time, it waited a second, and then it assigned that value. If I say trigger equals false, then the value of K is at assignment time. We fill in that assignment from there. So now K1 gets a value of 2.1 instead of 1.1 because we waited a second and time has moved on. We could even, like, this thing might make more sense if it was like we set it to the value of S1, right? So now we set it to 1.3 because S1 was at 1.3 at assignment time. And if we just change this to true, now K2 was 2.4 because S1 was 2.4 right here. So we waited a second before we assigned it, but we kept this value in memory before we actually gave it to the value. So there's a lot of flexibility here. And if you need to know something particular, you can look at the documentation or like email me and ask like, how do I do this crazy thing?
Oh,
It's probably available.
Somebody asked an interesting question.
Yeah.
So let's say you're running a long
yeah.
simulation, right? And you don't know how long you need to run it for, but you know that you've done enough simulation if, say ATP falls below 1 millimolar.
Yeah.
At that point, it would be nice if you could actually stop the run. Now we can't do that.
Right.
But it's an interesting idea though. Yeah, you can imagine having to run a long simulation and you don't know how long to run it for. Maybe you run it overnight, right?
Mhm.
And but you know that once something drops below a certain concentration, you just want to stop the run.
Right. You can do
Um,
that. You can do that on the tellurium side with
Yeah.
successive simulates. Like, because simulate picks up where it left off. You could say simulate for 100 seconds and then check. And if we're
well,
done,
we have a very specific alternative for that called one step,
yeah.
which is designed for that actually. So there's a
Mhm.
so you've got simulate, right? That does a continuous simulation over a range, generates lots of data points. We have another method called one simulate,
One
and
step.
that'll just run a simulation for a set step length, just a specific amount of time, then stop. So what you can do is you can put that one step inside a loop and keep calling it. And in fact, it returns the next time point as well to make it easy for you. So in the old days, before events were officially put into SPML, this is how we handled events. So you'd have a loop like this, and then you'd call something called one step. That would run a simulation for, say, 0.1 of a second, and then you'd check things to see what's happening. And if a certain thing had changed, you could then make the change then, and then the loop would go back and continue. Yeah, so that's exactly how it would work. Now the append, well, one step takes some arguments though, Lucian, if you remember.
Yeah, sort of like
It returns the
0.5 or
new time.
something. Does
It
that work?
returns, it returns the new time. So you have to give the current time and the step size. So you need to have you need to monitor your own time, okay?
Okay, yeah.
So you have to have a
let's see.
thing called T or whatever, set that equal to 0.
0.
And then in one step you have, I assume it's time, comma, the step size, and it returns time. It returns the new
Okay.
time.
So yeah, we want,
So you don't want to do any appending.
yeah.
Not there anyway.
Okay. So yeah, here we just do
So
that,
it returns
and
the
then we
new time,
say
so it's ready to go.
results append r.s1. Say
And don't forget to pick up the time it returns because it needs to be incremented. So in line 16,
well,
go
here,
time equals.
There.
Listen, you need to put the time
sorry.
equals in line 16.
Oh yeah. D equals
No,
to-
no. Line
Well,
16.
I need to increment time.
No, it does it for you.
No, but if I say one step t, don't I need the next time I call it to be t?
Yeah, but it returns the new time for you.
Okay, so I can just see. Okay,
T equals.
T equals that.
It was just a thing to make
Right.
it easy.
Got it. That makes sense.
Yeah, and that should in theory work.
Let's say 1.
So this was how we used to do events before we had official support for events in SPML.
There we go.
So I mean, it gives you a lot of flexibility being able to control every simulation step. So that's basically that.
Okay.
Yeah.
So yeah. So, or in fact, we should say there. So we actually start at 5 and then we go down.
Yeah, and you can collect the time as well if you want, of course.
Yeah.
So
as soon as it was less than 1, we broke out of our loop.
Oh yeah, that's
and that
it.
was
Yeah,
our last version.
right. Yeah, that's it. Yeah.
Yeah.
So there's a sort of a hack then, whoever asked, the sort of hack you can do to control the simulation time.
Yeah. It probably was somewhat slower.
Yeah, it would be slower.
Yeah, it's going to be.
I mean,
So yeah,
Python
you
is
getting
Python
your
is
time step
slow,
right is important.
right?
Yeah.
Python is slow, so now we do have Julia bindings. So you have Tellurium for Julia too, which would be much faster, but that's a completely different story.
Right.
All right, any more questions? Otherwise, I guess we move on to the next one.
Yeah.
Yeah. Great, thanks a lot for that.
Right.
Yeah, there's a lot of untapped potential, the whole event side of things, which I haven't explored much. I just use it for simple things, but I think you could use it for a lot more stuff, especially building hybrid discrete continuous models. I think that's probably a possibility. So,
Right. I'm going to stop the recording.
okay.